In vitro breakthroughs are redefining how we study the earliest moments of human implantation. Researchers have built a three-dimensional artificial uterus to mimic the embryo's natural environment. The goal is to recreate the soil where seeds, embryos, can take root and grow. Engineers tuned the hydrogel matrix to match the uterus's mechanical softness, measured by its Young's modulus. Beyond 2D cultures, a true three-dimensional environment brings the concept closer to reality. Microfluidic chips create separate channels to culture diverse cell types in concert. This allows epithelial cells, glandular cells, and supporting stroma to coexist like a real uterine lining. The engineered uterus provides a hospitable surface for artificial embryos to engage in a delicate dialogue. Researchers then introduce stem cell-derived embryo-like structures into the model. For the first time, the entire implantation process could be observed outside the body, from arrival to invasion. Key molecular players emerged as VAGFA and VAGFR1 direct signals at the embryo-maternal interface. Transcriptomic analyses revealed intricate conversations between trophoblast derivatives and endometrial cells. This model also serves to study implantation failures and test potential therapeutic compounds. For recurrent implantation failure, dozens of FDA-approved compounds were screened for efficacy. Promising candidates showed improved implantation rates in patient-derived endometrial tissues. Ethically approved donated human embryos were used to validate the model's predictive power. The team emphasized that artificial embryos are a model, like a flight simulator for aviation research. From cells to systems, the model integrates germ layer derivatives and tissue architecture. The research marks a shift from two-dimensional pictures to a holistic 3D reproductive landscape. This landscape enables researchers to explore genotype to phenotype effects in uterine diseases. Potentially, this technology could illuminate endometriosis pathophysiology and treatment avenues. Researchers published findings under the title A 3D in vitro model for studying human implantation and implantation failure. The shift from 2D to 3D mirrors the move from flat maps to immersive simulations in biology. The project's lead scientists include international collaborators who advance stem cell biology and embryo research. From deep tech to potential clinics, the researchers envision preconditioning patients' uteruses before embryo transfer. The ultimate aim is to enable successful pregnancies by tailoring the uterine environment to individual patients. This frontier also offers immense potential for regenerative medicine by deriving early organ precursors from stem cells. Ethical considerations remain central as the field advances alongside scientific breakthroughs. The work underscores why animal models cannot fully capture human implantation, underscoring the value of human-specific models. The team continues to collect diverse patient samples to understand the heterogeneity of recurrent implantation failure. In the coming cycles, targeted compounds may be used to precondition the uterus before embryo transfer. The project envisions turning a seed into a flourishing plant, an embryo growing within a supportive soil. If successful, the artificial model could accelerate discovery of therapies for infertility without relying on invasive trials. The researchers emphasize transparency and shared data to advance understanding across institutions. Ultimately, the work seeks to answer when and how the embryo first communicates with the mother's body. The research is grounded in biology, but it requires engineering ingenuity to mirror the complexity of living systems.